Hello, my name is Cameron King, and today we'll be looking at the digestive system, specifically the human digestive system, the chemistry of digestion, as well as catalysts and enzymes. So before we start, let's look at the word digestion. So what does it mean to digest something? To digest something means to break it down and then absorb it. And how do mammals digest their food? By using these specialized organs that are adapted to exactly this, breaking down the food into simpler molecules that can then be absorbed and used by our bodies. So let's look at the human digestive system. In the human digestive system, it is one long tract that moves food around using something called peristalsis. This is muscular contractions of the esophagus, stomach, and other organs to move the food through the various stages. Stage 1 would be the chewing. We call this mechanical digestion. This is the breakdown of food physically. There are, however, enzymes in the salivary glands or in the saliva of your mouth that also aid in chemical breakdown, and we're going to go through those later. Stage 2 is in the stomach. This is when hydrochloric acid as well as other enzymes are going to chemically break down the food. The food at this point is also known as a bolus. It is the bowl of food that is formed when you are chewing and you swallow. We call that swallowed food a bolus, moving down the esophagus by peristalsis into the stomach. In the stomach, there's also a form of mechanical digestion where the stomach will churn and move the food around to mix it. That's also breaking it down into smaller pieces. Stage 3 and final stage of digestion would be the movement of this food, also known as chyme, into the small intestine and then the large intestine. And this is where mainly absorption occurs of these nutrients and these broken down materials. But some chemical digestion also happens in the small intestine through enzyme processes. So let's look at some of these organs in more detail. So the esophagus, also known as the gullet, is the connection point between the mouth for mechanical digestion and your organs. The stomach, we said, contains hydrochloric acid. This acid doesn't only break down the food, but also kills any bacteria that might make you sick that could be on the food. It also is going to contain enzymes that will help break down the food molecules into a state where these nutrients can be absorbed. The hydrochloric acid is also going to create an environment that helps these enzymes function at their maximum capacity, and we'll look into that a bit later. So the small intestines, this is where soluble food molecules are absorbed into the blood. So there is a bit of absorption within the stomach, but when it comes to the small intestines, they are going to be more adapted and more suited to absorbing these nutrients into the blood through something called villi, which is what we'll go over later as well. So it has a strong blood supply in the small intestines and a short diffusion distance. So the distance between the blood supply and the actual food is very small, only a couple cells thick. Now your liver is going to produce something called bile. Now this bile is going to emulsify fats. What that means is fats have a tendency to clump up into little balls and stay there because they are hydrophobic. Now what bile does is it's going to break those balls up, therefore increasing the surface area and the amount of contact that there will be between the enzymes of the small intestine and the fat soluble molecules. And it's also going to contain something called a gallbladder. Now this gallbladder is going to be storing that bile from the liver for when the food leaves the stomach and enters the small intestine. What this bile does is it's also going to create a pH that is suitable for the enzymes found in the small intestine that may not work in the acidic environment of the stomach. The pancreas is going to produce hormones such as insulin to help with the breakdown of blood sugar, and it's also going to make other enzymes required for digestion. And the large intestine is mainly for the absorption of water and minerals. Okay. Now, what do we call these molecules that we are trying to break down? We will be referring to them as macromolecules. So one of the macromolecules are your carbohydrates. Your carbohydrates are there to provide us with energy. These carbohydrates include our starches and cellulose, and we refer to these big molecules as complex carbs. We use the enzyme amylase to break down these complex carbs into something our body can absorb and use. These are known as simple carbohydrates, such as your glucose molecules. Now, the test that we use to test for complex carbohydrates such as starch is iodine test, and the test we use for simple carbohydrates such as sugar is your Benedict's solution. Now, the next is protein. Now, protein is a macromolecule that can be used as a building material within the body. So it makes up our amino acids as well as our signaling in the body. Okay. These amino acids, we know from translation, join up to form these long chains. And these chains are then going to fold into a 3D structure that will eventually form enzymes. Enzymes are going to do a number of functions in the body. So protein is very important so that you can get these amino acids. Interestingly enough, there are some amino acids that your body can create itself, and there are other amino acids that we must get from a healthy, balanced diet. Okay. Now, these enzymes, which are amino acids that are folded, okay, can denature if exposed to too high of temperatures. This is why our body tries to keep a homeostatic and equilibrium temperature, okay, so that the enzymes in our body can function at optimum performance and not denature. Okay, but to make these amino acids, or to get them from the macromolecule of protein, we use the enzyme pepsin. Pepsin functions well at an acidic pH, so protein digestion happens mainly in the stomach. Okay. The last macromolecule that we'll be looking at is lipids. Okay. So lipids are used to store this energy that we get from our carbohydrates. All lipids are insoluble in water, meaning they don't mix well in water. And they are made of 3 fatty acids and a glycerol. The food test that we use for lipids is ethanol, and when lipids are present, ethanol gets a bit cloudy. And apologies, when it comes to protein, the food test that we use is the Burette test. When it comes to lipids, the enzyme that we use to break down those lipid macromolecules into the fatty acids and glycerol is something called lipase. Lipase functions within the small intestine at a higher pH. And then it's good to know as well that other than carbohydrates, lipids, and proteins, we also need vitamins and minerals. These we can get from our vegetables as well as eating certain types of meats. These vitamins are going to keep us healthy, and minerals are going to help us carry out other bodily functions. So the tests that we use to test for these various macromolecules are important to understand for your exams. You should know the positive for the test as well as the negative for the test. Okay. So when it comes to starch, we're going to use iodine solution. Now, iodine reacts with starch molecules and goes into this dark blue or black color from its original brown reddish color. Okay. So it goes from reddish brown into black in the presence of starch. If there's no starch, it will stay reddish brown. When it comes to fats, we said that ethanol is going to turn cloudy in the presence of fats. So it's usually clear and it goes cloudy in the presence of fat. When it comes to a test for glucose, the simple carbs, we use the Benedict's solution. The Benedict's solution is this light blue color, and depending on how much glucose there is, it is going to go from clear blue color, okay, into a reddish brick color. Okay, so if there is a little bit of glucose, it's going to be this gold color in between them. And when it comes to the protein test, we're going to use the burette solution. Now the burette solution is a sort of darkish blue color, and in the presence of protein, it is going to turn purple. In the absence, it's going to stay this blue color. So now that we've talked about the digestion and the breakdown of these macromolecules, we need to talk about how they're absorbed into the body. Most absorption of these smaller molecules that can be used by the body occurs in the small intestines. Now the small intestines are adapted to absorption through these appendages known as villi. These villi are hair-like structures that line the small intestine, and they increase the surface area and the amount of contact that the small intestine will have with the food molecules in the small intestine. There is a very small gap between the blood and the rest of the food that needs to be absorbed, okay, and a very large blood supply that we mentioned earlier. These villi are then also going to be surrounded further by microvilli, which are even smaller hairs, to maximize surface area and maximize contact with potential nutrients in the gut, and then absorb them into the bloodstream. Now we've spoken about the various enzymes of amylase, pepsin and lipase, but what exactly do enzymes do? Enzymes act as a catalyst. Now a catalyst is a substance that increases the rate of a reaction without being used up itself. So once an enzyme is made in the body, it can be used over and over and over for its specific function. Now, why does it only have one specific function? Because of the shape. The shape that an enzyme takes is going to take on the shape of the complementary substrate that it is trying to break down. We call this the lock and key theory of enzymes. Okay, so the substance of a specific enzyme, or the substance that gets broken down by a specific enzyme, is going to fit perfectly in the active site of that enzyme. And then the reaction can occur, as we see here, that splits that macromolecule into its smaller molecules at the active site of the enzyme. So previously we talked about the fact that the body keeps a constant temperature so that your enzymes can function optimally. Too cold and the enzymes are going to work slowly. Too hot and the enzymes can denature. Now if an enzyme denatures, it usually can't reform, and therefore its function won't be executed. Another factor that needs to be considered is the pH that the enzymes work in. Certain enzymes are going to prefer and work optimally and only work sometimes between a certain pH range. So the first is our amylase. Amylase breaks down our carbohydrates. This can be found in the mouth, where the pH is sort of neutral. Okay. The next is pepsin. So pepsin breaks down our proteins. We know that pepsin requires an acidic pH, and therefore it can be found in the stomach with the hydrochloric acid. Finally, there's lipase. Now lipase requires a basic pH. So we find that in the small intestine, and that pH is provided by the gallbladder producing bile. Okay, this has been the digestive system. My name is Cameron, and I'll see you next time.